Hi everyone, I'm Linxia Xiao, a PhD student from China University of Petroleum, East China. Today I will present my work, Automatic Identification of Sweet Spots from Microelectrode Recordings for electrodes implantation in deep brain stimulation of the subthalamic nuclear surgery. This work is done together with my colleagues in Shenzhen Institute of Advanced Technology, Shenzhen Second People's Hospital, the Chinese University of Hong Kong. This is the outline. Let's look at the introduction. STN-DBS is short for deep brain stimulation of the subthalamic nuclear surgery, which is the popular technique for the treatment of Parkinson's disease patients. During STN-DBS, optimal sweet spot is the position of electrode implantation for Parkinson's disease patients, which can achieve the best motor improvement. Thus, accurate identification of sweet spots is the key to successful surgery. The left picture shows the procedure of STN-DBS surgery. In the intraoperative process, clinicians detect the sweet spots with the microelectrode recordings, which is the significant clinical indicator for sweet spots identification. The right picture shows each part of the brain has its own electrophysiological activity. The sweet spots are determined by the microelectrode recordings with the high frequency and high amplitude features. Here are some difficulties for clinicians to identify the sweet spots. The clinicians try to implant the electrode in every sweet spot manually to find the optimal sweet spot from the border of post-STN to the border of pre-STN. Moreover, the intraoperative localization of sweet spots is a time-consuming and labor-intensive process. Currently, many models can be built to distinguish the entry and exit of STN with the microelectrode recordings. Some references demonstrate that a dimension microelectrode recording signal can be transformed into two-dimension Grammy angular summation field image to obtain the textual features. Moreover, attention mechanism can focus on the features of interest, such as the high frequency and high amplitude activities, and filter out the irrelevant neural activities information in the classification task. In order to overcome the identification problems of sweet spots in clinic, we should realize the automatic identification of sweet spots to reduce the dependence on surgeons' experience and shorten the duration of surgery for intraoperative sweet spots identification. Therefore, our work is to propose automatic and precise identification method of sweet spots from microelectrode recordings. The second part is methods. Here's an overview of our method. An automatic identification method of sweet spots from microelectrode recordings are presented for electrodes implantation in STN-DBS, including time series 2 image encoding and the attention-based convolutional neural network for sweet spots determination. Before experiment, we perform the processing for the microelectrode recordings data. First, we convert the microelectrode recordings to MATLAB data by map file converter and clip the entire microelectrode recordings by non-overlapping time windows of 1 second duration. In the stage of time series to image encoding, we convert the one-dimension microelectrode recording signals into the two-dimension GASF images. GASF is short for Gramian-Angular Summation Field. In this stage, we first normalize the microelectrode recordings and convert the microelectrode recordings in polar coordinates. Then we calculate the GASF matrix to generate the GASF images. Considering that the microelectrode recordings are inside the sweet spots or inside non-sweet spots, as shown in the picture, the generated GASF images from the sweet spots and from non-sweet spots has obvious bright stripe features and bright texture features respectively. Therefore, the identification of sweet spots can be defined as two-category image classification. In the stage of tension-based CNN for sweet spots determination, we adopt the convolutional block attention module enhanced networks to identify the converted GSF images by effectively integrating the CBAM into the CNN architecture. Our network consists of 7 stages and a fully connected layer. Each stage contains a convolutional layer and a convolutional block attention module. From the 4th to the 6th stage, a max polling layer is adopted at the end of each layer. The convolutional block attention module consists of 2 main submodules, channel attention module and spatial attention module. The reason for selecting CBAM enhanced networks is that the attention mechanism can better capture textual features and obtain more intuitive contextual information for improving the classification performance. The third part is experiment. In the experiment, we chose the microelectrode recordings from 100 patients to conduct the experiment. The data of 75 patients is for training. The data of 10 patients from training set is as validation set. The data of 25 patients is for testing. We conduct our experiment in MATLAB 2017 and Python keras. To evaluate the performance of our method, we first assess the effectiveness of feature extraction. The picture shows our method can enhance the stripe and the texture features and suppress the non-related visual image features, which is helpful for identifying the sweet spots from microelectrode recordings. Then we evaluate our method from qualitative and quantitative perspectives. As shown in this figure, the horizontal axis is the time, the vertical axis is electrode implanted depth. According to the neurosurgeon's decision, the green line indicates the border of pre-STN. The blue line indicates the border of post-STN. The microelectrode recordings from sweet spots are labeled in the black rectangular boxes. The result of our method is shown in the right picture. The micro recordings from sweet spots are identified and labeled in the black rectangular boxes. By the analysis of two above results, The identification results of our method is consistent with the results of new surgeon's decision. The table shows the accuracy of our method is 96.72%, which outperforms the state-of-the-art methods. The precision of our method is 98.97%, which can fulfill the clinical precision requirements. In this study, we propose automatic identification of Swiss balls from microelectrode recordings for electrodes implantation in STN-DBS. Experimental results shows our method is consistent with the clinical decision-making, and attention mechanism can improve the precision of Swiss balls identification. However, there are two limitations of our method: data from a mechanical center and one equipment, and the lack of a strategy of the optimal target spot selection. In the future work, we will collect data from other sites and equipment, and will employ multi-modality data fusion technique to define the sensory motor region. Thank you.